Hey guys, Mr. Gibson here. So I'm recording a video. This is for going over the Unit 2 study guide. So hopefully at this point you have finished the study guide and you are just going through this and watching this video to make sure that you got the questions correct and correcting any ones that you may have gotten wrong. Okay, this is not meant for you to fill these in as you go for the first time. Okay, this is just correcting answers. So let's go ahead and get started. Scientists believe they've discovered the gene that codes for the protein hemoglobin, which is responsible for binding oxygen in your red blood cells. What question or questions would help them gather more information regarding the relationship between this gene and hemoglobin? So some people are going to be tempted for this to say, well, let's find people who have the hemoglobin or functioning hemoglobin. Find people who their bodies are making hemoglobin and see if they have that gene. Here's the problem with that question. That you have lots of genes and you have working hemoglobin. Okay, saying that you have working hemoglobin and you have this gene you think codes for hemoglobin doesn't provide evidence that that is the correct gene. It could be any of your working genes. So what you want to do is find out, why is that highlighted blue? Find out, does someone without that gene, or with that gene being miscoded or a mutated gene or something like that, without that gene, have trouble finding oxygen in your red blood cells? Okay, because that's what hemoglobin does. It binds oxygen, transports it in your red blood cells throughout your whole body. So if you can find someone without that gene or with a miscoding in that gene, and you test them and you see, oh, they don't have functioning hemoglobin. This gene is messed up and hemoglobin's messed up because they're not transporting oxygen correctly. Then that would give you a clue that there is a relationship between that gene and hemoglobin. Okay. I think I'm going to switch my font color to red so it stands out. Okay. Explain what determines why cells from the same organism have different functions. Okay. Well, proteins regulate which genes are activated and which are not. Remember that diagram I showed you where you had, the heart cell, liver cell, skin cell, eyeball cell. I don't remember all of them, but you had these different cell types and they all had the same genes. All cells in the same organism have the same DNA pretty much. So what makes them different? What gives them different functions? Well, it's the combination of which of those genes are turned on and off, because that will change which proteins are made, and proteins determine cell function. And proteins, repressor proteins and activator proteins, are the ones that regulate or turn on and off these genes. Okay? What is the role of the CFTR gene in those who have cystic fibrosis? So the CFTR gene, CFTR gene regulates the production of mucus in the body and digestive enzymes. Okay, so it regulates the production of mucus, specifically in your lungs, and digestive enzymes. So it also has to do with sweat glands too. But if someone who has CFTR, who has the normal CFTR gene, they've got normal mucus production, normal digestive enzymes as far as that gene goes. Someone who has a mutated version of that gene has the disease cystic fibrosis. Okay, so that means this gene is mutated. I meant to say CFTR gene, meaning that they're going to overproduce mucus and their digestive enzymes are going to be all out of whack too. Okay, and those are the symptoms of cystic fibrosis. This says, describe the relationship between genes, DNA, and chromosome. Well, genes are segments of DNA that code for proteins. Chromosomes are a single strand of DNA coiled around a histone protein. Think of that as like the middle of the paper towel roll, okay, that cardboard piece. The DNA is wrapped around it, and that's what forms a chromosome, okay? And then sections of that DNA strand that code for proteins, those are what we call genes. So what is the function of chromosomes? Well, generally, the function of chromosomes is to regulate the production of proteins. Okay, think of chromosomes like a cookbook. Okay, proteins would be the food that's made. So the chromosomes hold all of the information, all of the recipes. They regulate which proteins are going to get made or produced. Okay. What factor determines the trait that a gene codes for? Well, when you get down to it specifically, it's the sequence of nucleotides in that gene. So those A, T, C, and G, the sequence of those letters is what determines the trait that that gene codes for. So those are like the steps in the recipe. Okay, so if a gene is a recipe in the cookbook of your DNA, okay, it's a recipe for one protein. The steps, the sequence of how to make that protein is found in the instructions in the recipe, which in this case is the sequence of those letters, the sequence of nucleotides. Okay, if you want to change the protein, you change those letters. Explain the role of DNA and chromosomes in passing genes to offspring. Well, just to put it very generally, okay, each parent passes on 23, this is in humans, chromosomes. We call these haploid cells, the ones that only have half the normal amount. In all species that reproduce sexually, they pass on a haploid cell, or they pass on a sex cell that only has half of the normal amount. In humans, that happens to be 23 chromosomes because the normal amount is 46. So each parent passes on 23 chromosomes to the offspring. And genes for the offspring's traits are carried on those chromosomes. Okay? So, yeah, that's generally what's going on. You get 23 chromosomes from your mom, 23 from your dad. That gives you 46. And then all the genes, all the information for how to make your proteins and specific cell types and all that stuff come from those chromosomes that you inherit. What are possible functions of non-coding DNA? So most of your DNA, actually about 90% of it, is not genes. Okay, it's non-coding, it's non-regulating. Okay, so the question is, if it's not genes, if it's not used to make proteins, what's it used for? Well, it can be used for a couple things. It can be used to help the structure of DNA. Okay, it could be used to protect the DNA. So you've got some at the tail end of your chromosomes, you have what's called telomeres, and it's just a long sequence of non-coding DNA that protect your DNA every time it gets replicated. But it's also serves as binding sites for the proteins that do DNA replication and transcription. Okay, so those proteins who copy the DNA either to make more DNA or copy down the DNA into an RNA strand so that it can go make a protein. Those proteins have to know where to attach. They have to know where to start reading those genes. And so maybe the non-coding region in front of it, called a promoter or a primer, depending on which one of those two processes we're talking about. That's kind of like the landing strip for the protein. Tell, hey, land here, and then start reading the gene. Okay. This is diagrammed the process of meiosis. So let's just do that really. Really simply, let's insert an image right here. Okay, so insert drawing. Okay, so very simply, check this out. you've got one cell that divides into two cells. Okay, move these guys around. Ooh, that was weird. Okay, and then those two cells then divide into two more cells. So it goes through PMAT, like in mitosis, but it goes through it twice. So it goes through PMAT once, and then it goes through PMAT a second time. It's a little bit meticulous. So now I'll just copy this, okay? So this is probably the fastest way I could think to diagram it. Okay, so this starting cell is what we would call a germ cell, and it starts out diploid, meaning it has 46 chromosomes, full amount, and then after it's done dividing, all four of these cells at the end of meiosis that we call gametes or sex cells are now haploid. They have half the amount, and they need half the amount because they're going to meet up with another sex cell at fertilization, and that way the offspring has the full amount. Okay? If this was a male, all of these would be sperm cells, and if this was a female, this process is used to make egg cells. Actually, only one of these would become an egg and the other don't, but we're not going to get to that complicated business, okay? So I'm going to save that, insert it there. I'm going to shrink it a little bit because it's a little big, okay? And there we go. There's meiosis. Cool. How does meiosis contribute to genetic variation? Well, there are two ways. The first way is crossing over. Crossing over is when homologous chromosomes, or chromosomes that are very much alike each other, they have genes in the same location and they actually line up next to each other during meiosis. So homologous chromosomes swap genetic information during, it's actually prophase 1. Okay, so that's one way. This random swapping of genetic information while the chromosomes are dividing really kind of mixes up the genes that adds to the variation of the genes. The other way is independent assortment, which is the random sorting of chromosomes into sex cells at the end of meiosis. Okay, so it's random which chromosomes are going to go, which versions, which different alleles or versions of genes are going to go to each chromosome, or going to go to each sex cell at the end of meiosis. So we call that independent assortment. If you cross two heterozygous parents, what's the likelihood the offspring will exhibit the positive trait? It says show me in a Punnett square. You don't have to if you can do it in your head. So two heterozygous parents, you're going to have the likelihood the offspring would exhibit, and it shouldn't say positive either. Glad I caught that. It should say dominant. Remember that from last year now. So if you remember, the phenotypic ratio, meaning the ratio of dominant phenotype traits being expressed and recessive traits being expressed, whenever you cross two heterozygous parents, it's always 3-to-1. So 3 out of 4 would be 75%. There's a 75% chance offspring will exhibit the dominant trait. If you are fuzzy on that, work it out in a Punnett square, okay? If you can do it in your head and get it right, go for it. Provide evidence from a real-life scenario that environmental factors can cause an increased risk of mutations. Well, the main one I can think of is skin cancer caused from too much sun exposure, specifically UV radiation. Okay, so like if you go to a tanning bed and you sit there for like 6 hours, that UV radiation, that high intensity UV, can actually cause skin cancer. If you spend too much time in the sun, especially in like a warmer climate closer to the equator, and you don't wear sunscreen, you're exposing your cells to UV radiation, which can mutate the DNA, causing cancer. Okay. If a tongue roller and a non-tongue roller had a child, what is the highest possible percentage of the offspring who would be tongue rollers? Tongue rolling is dominant. So it depends. So if tongue rolling is dominant, then the non-tongue roller is recessive, meaning that person is going to be homozygous recessive or have two lowercase alleles, two recessive alleles. Okay, we know that. So if the tongue roller who has a child with the non-tongue roller is homozygous dominant or has two dominant alleles, then there's a 0% chance that the offspring would be a non-tongue roller, because they would always inherit that dominant allele from the tongue rolling parent. But if that parent who can roll their tongue is heterozygous, meaning they have the dominant allele, they show the dominant trait, but they also have the recessive allele, they just don't show it, then there is a 50% chance that offspring would be a tongue roller and a 50% chance they would be a non-tongue roller. And that's the highest possible percentage, because in the other scenario, it's 0. Okay. If that was confusing to you, we'll work it out in a Punnett square. Okay. Explain how crossing over can result in increased genetic variation. Well, those homologous chromosomes, it's really just looking for you to be able to define crossing over. Those homologous chromosomes are exchanging genetic information during meiosis, okay? So that is what increases, that's how it increases genetic variation. It says, describe the factors that have the highest impact on increasing genetic diversity. There are 3, okay? Crossing over, we've heard that like 3 times now. Crossing over is one of them. Independent assortment, those are the biggest ones. Okay, there's another one, but it's rare, but it does add to genetic diversity, and that is mutations. Okay, so you got crossing over, the random exchanging of genetic information during meiosis, independent assortment, the randomness for how those chromosomes get separated out into those sex cells, so it's random which ones you're going to inherit, and then mutations, it's just the random changing of DNA through some kind of miscoding or something like that. Okay. Finally, no, not finally, JK, we got 2 more. So 2 colors of flowers, red and yellow, are bred together. P1, the parent generation. All of the offspring F1 are red. Explain how this is possible. Well, if the red flower is if the red color is dominant, if that's the dominant trait, yellow is recessive, it is possible to breed those 2 colors and have all red offspring if the red parent was homozygous dominant. If it had 2 dominant traits, then it would always give a trait, or 2 dominant alleles, it would always pass on an allele that would overshadow that recessive one. Okay. It says, then describe the results if the F1 generation is self-pollinated. So you take that F1 generation, that first generation of offspring, who are all heterozygous now because they got 1 dominant from the red, 1 recessive from the yellow, and you cross-pollinate them, then, well, first of all, I'll just say in P1, all the red were homozygous dominant, all the yellow were homozygous recessive. Okay? F1 are all heterozygous, all of them. Okay? If you don't believe me, work it out in a Punnett square. So F2, or the generation that we get from breeding F1 with itself, it's self-pollinated. So you're taking, basically have 2 heterozygous parents. Okay, so that means the genotypic ratio would be 1 to 2 to 1. If you work that out in a Punnett square, you would have 1 homozygous dominant, 2 heterozygous, 1 homozygous recessive. Those are your genotypes. Okay. The phenotypic ratio, however, you would have, because you have 1 that's homozygous dominant, 2 that are heterozygous, that's 3 offspring that are going to show the dominant trait. So it's going to be 3 to 1 for the phenotype ratio, or the ratio of red to yellow flowers. Remember, the genotype is the specific alleles they have. The phenotype is the trait that is expressed. So in this case, it's flower color. All right. So it says, provide a rebuttal to the statement of two parents with dominant traits. So this needs to go. I'm going to delete that actually. It's a typo. Okay, how do variations of traits develop within a family? For example, why do members of the same family have different traits from each other? Answer with a CER, provide a rebuttal, which is kind of just like a counter argument. A rebuttal in your reasoning discussing how parents with two dominant phenotypes can produce offspring with a recessive phenotype. So my claim would be, I'm not going to actually type this out for the sake of time, but my claim would be members of the same family can have very different traits from each other. Boom, there you go. Now let's prove that. You're going to talk to me about how that's possible. Well, what are the three things? What are really the two big things that increase genetic variation within a family? Crossing over and independent assortment. Happens every time meiosis happens. Okay. Now, if you want to talk about mutations, you can. But crossing over and independent assortment are really those two big things. Okay. So I'd maybe just define those as the evidence. Okay. And then in your reasoning or even evidence, you can just list those words. Okay. And then in reasoning, you're going to explain how crossing over causes family members to have different traits, and how independent assortment causes family members to have different traits. And then we got to look at our rebuttal. So how could parents with two dominant phenotypes produce offspring with a recessive phenotype? So let's take cystic fibrosis, for example. How can two parents without cystic fibrosis have offspring that has cystic fibrosis? Well, if both parents are heterozygous, then they both carry the recessive gene, the recessive allele. Okay? They don't show the recessive trait because they're heterozygous. They also have that dominant there. Okay? But if they were to have an offspring, there is a 25% chance when you cross two heterozygous parents, there's a 25% chance that offspring will inherit one recessive allele from the mom, one recessive allele from the dad, and therefore produce a recessive phenotype, even though neither parents showed that phenotype. Okay, so that's how you would type out that CER. If you guys have any questions at all about this content, feel free to email me. Yeah, if you guys need any help at all, let me know. All right, good luck.